Tell you something about the structural analysis of glycans with the focus on CDGs. I came here from Slovakia, from Bratislava, that's the capital city. Slovakia is a small country. We have 5.5 million of citizens, and every year we have 53,000 of births. I am from Center of Excellence for Glycomics, so I am not the clinician, not the psychologist, but I do my PhD in analytical chemistry. This center was founded in 2010. Sorry, that's the mistake. And the center is focused on glycoconjugates and analysis of their structure.
Can you get closer to the mic? You're hard to
Sorry.
hear.
Sorry.
Thank you.
So the glycosylation of proteins in general, it's changed in cancer, in neurodegenerative diseases, in immunological diseases, and of course in CDGs. Diagnostics of these diseases in Slovakia was established in 2012 when the selective screening was established based on isoelectric focusing of transferrin. I would say most of you know this method. And up to this date, we tested about 1,000 suspected patients, and 3 individuals manifested abnormal transferrin isoelectric focusing pattern. The polymorphism was detected in 5 children, and the secondary causes of abnormal glycosylation were excluded in these cases. But for further solving of a specific subtype, the cooperation between hospitals and research institutions is essential. What's the glycoprofile? In general, we can say glycoprofile means all the glycan structures found in your serum. Everyone has its own glyco fingerprint. Glycoprofile, it's dynamical. It could be changed in 2 principles. You can observe minor changes, for example, when aging or when you get some infection, and you can observe major changes that are typical for more pathological conditions, such as already mentioned cancer, when the sialylation is increased, and of course in CDGs, when the single step in the glycan synthesis is disrupted. This pathway, I would say, probably most of you know. Just very briefly, it starts on the lipid-linked carrier. The first N-acetylglucosamine is added. The second one, the pathway continues. And when the glycan looks like this, it is co-translationally transferred to the nascent protein, and then it's transferred and processed. The final structure is released into the human serum. There are more than 80 known subtypes due to specificity of glycosylation enzymes and transporters involved in this pathway. What we can do? We can analyze this structure, a lipid-linked oligosaccharide, before it's transferred to the protein. So it's called analysis of lipid-linked oligosaccharide. This one is membrane-bound, and it's typical for analysis of CDG1 subtypes. And we can do analysis of secreted glycoproteins, and this is useful for CDG2. Isoelectric focusing, it's a good method that can separate transferrin isoforms in the term of numbers of sialic acids. But this method cannot provide any information about how the N-glycans looks like. So we are unable to determine a specific enzyme or the transporter defect. So the structural analysis is essential. Maybe some of you already know this picture. How does defocusing looks for healthy 1A, and this was just an example of a patient. What's our workflow? So, as I told you, we can do 2 types of analysis. So the first one is analysis of lipid-linked oligosaccharide. We can start with any sonified cells or tissue or anything that has Golgi apparatus inside. We do the digestion, we extract these allelose, reduce them, we release the oligosaccharides, and then we do the cleanup, and this is the result. So the final LLO should look like this. Another workflow is a little bit more complicated. We can start again with any sample that is serum or any body liquid. or just pure glycoprotein. Again, we do the digest at first. We isolate the glycopeptides, then release just N-glycans. We do the fractionation. We got a neutral fraction and sialylated fraction. We clean them. We label them to enable the quantification. And the final approach is LCMS. We use Hayek's column, and we use Moldy-Tofto spectrometer. What can help us is a database. We have so many databases to search and compare our spectra with. And we can also confirm the linkages by NMR, by specific derivatization methods. They are available, and by specific exoglycosidase digests. We got a lot of spectra, and per one person, it's more than 1,000 spectra. This is just to show you how it looks like. The lower line is a chromatograph from healthy patient or healthy individual, and the upper one is from the CDG patient. What is changed? In this case, you can very nicely see this one and this one. In here, we observed increased acialo and hybrid structures, and in this case, it was completely absent tri- and tetracellulated structures. Although antennae were built correctly. So we started with 80 possible defects, and now we reduced the possibilities up to, let's say, 15. So, but to continue the mutation analysis or another approach to complete the study is essential. Just to show you in here, this is how it looks like. So every single peak from the chromatogram is analyzed by Moldy-Tofto MS-MS. So you can get the information about the quality, if this is really in this peak. So I would say the combination of mass spectrometry and HPLC chromatography enables quantification as well as determination of every single structure. Maybe a lot of you already know this picture. You can see increasing number of CDG subtypes identified, but I would say the number of known subtypes, it's increasing not because these disorders have not been existing so far, but because of still more sensitive analytical technologies available. So just to conclude, I wanted to say just the cooperation between research laboratories and hospitals is essential in evolution of further diagnostics. And completing the informations about the structures can help us to understand the molecular basis of these disorders. Thank you very much for your attention.